Welcome to the world of prokaryotes. Prokaryotes are incredibly numerous, exactly what the slide says. They outnumber all of the eukaryotes in the entire world. Remember, the eukaryotes are the ones who make up all of the other kingdoms. This one on the right-hand side is a thermophile. This is one that lives in a very hot water vent at the bottom of the ocean. Prokaryotes can adapt to many different places. So we have archaea and we have bacteria. Those are two types of prokaryotes that are going to exist in archaea. And then we also have the regular bacteria. Those are our two main prokaryote domains. We have archaea and we have bacteria. Archaea, there are two main ones right here. There are the thermophiles and the halophytes. The thermophiles, like the one over here on the right-hand side, they live in the hot places. The halophiles live in the salt places. And then there's another group, and those are the methane makers, and they can live in the guts of the animals or, the craziest of rotting places. So prokaryotes are incredibly small. Their collective size is small, but their impact on Earth is humongous. So yes, they can carry diseases, make diseases, are diseases, but we also need those prokaryotes. We need bacteria. They decompose many different things. They help get rid of different things in the ocean. They break things down. They can cause illnesses for sure, but they also return incredibly awesome vital chemicals into the environment that we need. So let's look at this. This one says 5 kingdoms of classification, and I can understand it this way, and I hope maybe you can too. So here is the complex multicellular eukaryotes. So we have fungi, we have plants, we have animals. And in this lesson this week, we're not going to focus as much on plants and animals, but we are going to focus on prokaryotes, what I'm discussing with you now. We're going to call them right now, real quick, Monera. We're going to talk about fungi in a little bit and protists. So, like I said, there are 3 domains. Well, prokaryotes make up bacteria and the archaea. So we have the heterotrophic bacteria, we have the halophiles and the thermophiles, and then again over here in eukaryota, we have all of the other things like the protists, the animals, the fungi, the plants, and then there are some other ones they are thinking about adding into that phylum, but they haven't quite decided what they want to do. So what can prokaryotes do? Well, there are many diseases that are listed here that are bacteria. There's anthrax, the bubonic plague that you have talked about before. Many of us have heard about Lyme's disease or know somebody. Our dental cavities, ladies and gentlemen, are bacteria. If you've ever seen or scratched your teeth and you kind of feel that film that goes across, like if you take your tongue around your teeth and you kind of feel that yicky stuff, that's actually bacteria that's eating at the sugar of the food that you eat. That's the bacteria that's eating off of that. So if you're not brushing your teeth, that's actually the bacteria that's trying to eat off of it. Typhus is, if you read about it, it causes a headache and a high fever. All right. How is it transmitted is also over here. The type of bacterium is here, and this is the description of the disease. So the bubonic plague, well, let's find out. It says fever, bleeding, lymph nodes that form swelling called bubobs, and then often it is fatal. So the bubonic plague, if you've ever heard of it, which you have, that was something that a lot of people had died from. Well, what had caused it or transmitted it? The bite of an infected flea. Nobody wanted to be bit by fleas, but it did happen. If you look down here, here was another one bit by an infected flea or louse. Here's another thing that was carried by an insect, okay, or type of arthropod. pod. So you have to be very careful when you're out in nature because there are things out there that you might not want to get bit by. So they are unicellular. Many of them have what's called a flagella. It's kind of like a tail that whips back and forth. Some of them have capsule structure or slime layers. So it helps to allow them to invade the immune system and adhere. They're kind of like sticky or pilly. They have hair-like projections. It's kind of interesting. And then there's this fact right here. The E. coli is a 4-micron bacteria, a common symbiotic found in the digestive tracts of animals. So let's quickly look at this cell structure of a simple prokaryotic cell. So here's the capsule part. It's got cytoplasm. We've talked about it. There is a nucleoid. Okay, here's that tail that I call the flagella that allows it to whip around. Right here are the pili that come around on the outside. All right, there is no there is no organelles there. So we've talked about how there's going to be a major difference. Right? Now we can group them many different ways. We can group them by, is there a cell wall staining? We can group them by their shape and how they get their energy. So let's look at the bacteria cell wall. Is there one that looks like a cell wall? Is that unique material there? Well, if it is, it's a pepto. You know what? I can't even say that word. You tried it home. All right, and this creates a single molecular network that encloses and protects the entire cell. Eukaryotes don't have that. So when you look at this cell again, this structure, here is that extra cell part like a cell wall. You can see the tiny ribosomes that's right there. Now, we can look at these shapes. So we have the round shape, the coccus. We have the rod shape, the bacillus. And then we have the spiral shaped, the spirillum that's wiggly. Some will form into clusters like a staff, and some will fill into chains or filaments like strep. All right, so think of strep throat. So when we look at a picture, we can see how they can get into the clusters as it was just talking about. So you can see how the bacteria is going into the certain shapes. So the bacillus, the coccus, and the spirillum. So here's an example of what strep throat can look like under a microscope when you have all of them coming together into a chain. Here is another example. This one is spirilliform bacteria with flagella found in a rotting carcass of a bryozoan colony. Ooh, here is a rod bacteria living attached to a strand of filamentous algae. So the algae is actually the long part that has green, and the small parts that are coming off of it, that is the actual bacteria. So how do they get energy? So we talked about autotrophs already. We talked about heterotrophs. So autotrophs make its own food, photosynthetic, and then there's also chemosynthetic. So the sunlight, we know, allows us to make our own food if we're a plant. And then there's inorganic compounds. That is brand new to us. And then there's heterotroph. They get its food from other sources. So we can look at these heterotroph photosynthetic ones right here, and you can see there is something specific in common. They all have the color green. So here is cyanobacterium, okay, and it's probably going to be able to make its own food somehow. So it's going to be an autotroph, and that's simply because it's green. So when we're down in the bottom of the ocean with these hydrothermal vent communities, we're going to have some of the autotrophs because they don't have the ability to see the sunlight. So there's going to be something down here that's going to give the chemosynthetic autotrophic ways of getting the energy by themselves. which I think is incredibly awesome. All right, so then we also have bacteria that is going to, oops, let me go back to that picture, that is going to be what we need so that we don't walk around with dead carcasses all over the place. So here we see this really fantastic food web, but now our bacteria is down here, and you can see a microscopic blown up version here. It's breaking down all of the particles that has been used over and over and over again, from the bird to the snake to the mouse to the oak acorns and the salamander and the pine and the pine borer. Those are all things that eventually will be eaten, or will eat something and have died, and then will need to be decomposed, whether it's by the bacteria or the fungi. All right, let's talk about some interesting facts real quick. All right, most of what we know about metabolic pathways we learn from prokaryotes. Those, again, are the bacteria. And E. coli is the best understood of all of these organisms. All right, bacteria. It is so important. And I bet you probably didn't know all of this information about its food part and its chemical production part. It can make a lot of food. If you like pickles, olives, vinegar, sauerkraut, some sausages, those are all within some yogurt even. Those have bacteria parts in them. And pharmaceutical companies culture bacteria to make vitamins and antibiotics. The food industry, you need it for milk, ice cream, those kinds of things, cheeses. We need to have bacteria in order to make them. Mining in the environment, oh my gosh, if we didn't have bacteria in the environment, that would be awful. Okay, we need them because they help cycle all of those things, as I said, within the food web. The term biodegradable is used to show how bacteria eats away at these materials. We have different types of bacteria for sewer treatment, for them to synthesize petroleum for food, as I said way back earlier in the beginning of the video, to clean up oil spills, and they can decompose pesticides. So that is even really cool. Here's just a quick slide to show the ecological importance with the nitrogen fixation, and here is how bacterial toxins can come into play. They are good and bad for humans. So we all know that COVID-19 is going around now. We'll talk about that in a little bit. But some of the exotoxins are, botulism or salmonella, you know of those. We could use it for biowarfare. That would be incredibly dangerous. There are also endospores. Some bacteria create endospores. So we have to be careful, because if they're left in a non-hostile condition, the cell can remain dormant and then they could come back. And if they come back, it doesn't always mean that they will be killed. So boiling water cannot kill most endospores. So canned food industries have to take extra precautions. So you have to see if the can is bulging out. We do not want to eat it. We do not want to. All right, so how do they multiply? How do we know what happens? All right, well, binary fission is one thing in particular that we have to understand. We've talked about it a couple of times this school year. So it's an asexual reproduction. We have the prokaryotic cell, and it has the DNA on the inside, and it copies itself. And then it simply splits into two right down the middle, and then it has two identical haploid cells. That's pretty easy. But then there is kind of three other ways in order for bacteria to separate itself. There's conjugation, there's transformation, and there's transduction. So that helps with the diversity in bacteria. And when you see this, it is kind of like a sexual reproduction, but it's not. So we have a transfer of genetic material between two bacteria cells. So that is one way for it to happen. So in conjugation, this part right here, these are called plasmids. And here's our bacterial DNA. So the small circular self-replicating DNA molecule separates from the bacteria's chromosome. All right. And then what happens is that the conjugation DNA transfer is only one way. So it's going to go from one to the next. So when you see that the mobile plasmid is here, the pilus is here, the arrows are going, all right, and then you see how this has unraveled, and the new donor is here, this is the bacterium, and then it's going to transfer. All right, don't get that confused with transformation. They are genes that are taken up from the surrounding environment. All right, so here's the DNA fragments from donor cells. Here is the bacteria. The recipient cell takes up the donor DNA. So here's the environment right here. And the bacteria is here and it soaks it up. Okay? And then as it soaks it up, it kind of goes into the DNA. It's genetically transformed. And then the third one is transformation. And then it's finally transformed. The third one, is transduction. This is when the genes are transferred between prokaryotes by virus. So here is our little virus. It kind of looks like a space shuttle. So the space shuttle virus kind of attaches itself into the bacteria. It shuffles things all around. It gets all in there. They're called phages. And then they come around and then it leaves out of the bacteria only for the bacteria to now have its DNA compromised. And as it's compromised, it is now different. Because if you would have seen before, it had something different from the beginning to the end. So that is its difference. Okay? So what happened was this right here, this virus, was absorbed. So transduction was through absorption. Let's get into some of those antibiotics. So Alexander Fleming was the person who discovered penicillin, and he noticed mold growing in petri dishes. It was bacteria did not grow where mold was. He isolated the chemical that killed bacteria, but it was not stable. And Howard Florey continued the work, and he later stabilized that chemical. And they received the Nobel Peace Prize, or the Nobel Prize in 1945. Here is an example of Bacillus subtilis, a rod bacterium from the soil. It produces the antibiotic bactracin. Here is Streptomyces, and it's an elongated bacterium used in making a lot of antibiotics. And here, let's talk about it real quick. Antibiotics are substances that are produced by microorganisms, usually bacteria or fungus, that kills or slows the growth of pathogens. All right, so it's medicine. It assists the white blood cells because those are your fighting blood cells, and they help to fight the infection. And only bacteria or fungus, it only kills the bacteria or fungus. It doesn't actually kill viruses. You can't kill a virus, which is why we're having a hard time with the COVID-19. So the antibiotics does not work for viruses because they have a different cellular process, such as a cell wall formation and protein synthesis. Viruses do not undergo cellular processes. They do not affect them at all. All right, so how does an antibiotic work? The chemicals will attach to the cell wall of the bacteria and break it apart. So there's a lot of issues with antibiotic resistance right now. If you decide, or other people decide, that they need to have an antibiotic right away because they don't want to be sick, There are people who are unnecessarily using them and they're misusing uh, antibiotics, and then those antibiotics become so much in the environment that the bacteria is actually becoming resistant. And once the bacteria becomes resistant, then the antibiotic no longer works. So it says it is estimated that 50% of these latter prescriptions are unnecessarily because they are being prescribed for colds, coughs, and other viral infections because they are no longer being decently used against the bacteria, since the bacteria is like, oh, hey, I know what you are, and you don't affect me anymore. So, ladies and gentlemen, that is it about archaea and bacteria and everything that you need to know about those two domains that break down into Monera.